Hello students, this is Dr. Mohammad Zafar Iqbal. I am from Government Art Science College, Karwar. And in this video, this is a video on genetic engineering. And in the first video, we are going to discuss about the DNA extraction. This is the first very important thing which is necessary in case of all the experiments of the genetic engineering. That's why today we are going to discuss about the extraction of DNA from the animal as well as from the bacterial cell. This is the part of this particular video. And before we get into this topic, we are going to set some learning objectives. The first learning objective is to how to extract the DNA. We are going to learn how to extract or isolate the DNA, genomic DNA from the cells of an organism. Second, we are going to study about the how to analyze the quality of the that is the quality to analysis, that is the quality of the DNA molecule. Once we have extracted the DNA molecule, we don't know what is the quality of that DNA molecule, and that we are going to study how we are going to analyze the quality of the DNA molecule. Then we are going to study about the quantitative analysis of the DNA molecule, because in any genetic engineering experiment, you need to have minimum quantity of the DNA molecule. If the DNA is very less, then it may not be of any use. And that's why in this video, we are also going to discuss about how to analyze the quantity of the DNA molecule. What is the total amount of DNA present in a per ml that we are going to study. These are the three important objectives which are set for this particular video. And in this, first of all, if we would want to study the isolation of the DNA molecule, we are going to, generally, we study three different types of the DNAs. One is called the genomic DNA. This is nothing but the DNA which is present in case of all the organisms. This is called the DNA which is present in case of in the nucleus is called the genomic DNA. And this is since it has got all the genome, that's what is called the genomic DNA. The second type of DNA which is generally used in genetic engineering is called as the plasmid DNA. This is the second type of the DNA. Here, if you want to use only plasmid, there is no problem, because plasmid itself is a DNA molecule. That's why, just for the convenience of the student, I have written plasmid DNA, but plasmid is nothing but itself is a DNA actually. Okay, and the third type of the DNA which is necessary in case of the genetic engineering is the bacteriophage DNA molecule. And why we are categorizing these three? Because if you take the methodology of the isolation of the DNA, there is a different method for the isolation of the genomic DNA. There is a slight modification in the technique for the isolation of the plasmid, and there is a slight modification in the extraction of the bacteriophage DNA. That's why these are the three types of the DNA molecules which are generally used in recombinant DNA technology or the genetic engineering. That's why that's how I am going to highlight these three. And in this video, we are only going to concentrate on the isolation of the genomic DNA, because the extraction of the genomic DNA is very important. And that's why in this video, we are only going to discuss about the isolation of the genomic DNA. And for the isolation of the genomic DNA molecule, we need to have the tissues. From which tissue we are supposed to extract the DNA molecule. Take, for example, so many different types of the tissues we have in our body, and you may choose one or two type of the cells from your body, from a particular organism, to extract the DNA. That we have to choose which tissue you want to extract the DNA molecule. And second important thing is that the DNA can also be extracted from the bacteria. There is a slight difference. If you take the tissue preparation, in case of the tissue preparation, it is a solid tissue, whereas in case of the bacterial DNA extraction is slightly different, because you have to grow and harvest the bacterial culture. Then only you will get you can isolate the DNA from that particular bacteria. And the second step is called the preparation of the cell extract. Now you have the tissue for the extraction of the DNA molecule, and now you have got the bacteria, or the bacteria for the extraction of the DNA molecule. Next step is the preparation of the cell extract. Now you have to lyse open the cell, so that whatever the content of that particular cell is there, it should be released into the media, and that particular process is called the preparation of the cell extract. This is also a very important step. Unless and until if you don't break that particular cell, then you are not going to get any content of that particular cell actually. That's why you have to break open the cell, and once the cell is broken into pieces, then whatever the extract is there, that particular extract will have a DNA as well as other biomolecules. And next step is called as the purification of the DNA from the cell extract. And I have already said that when you take a cell extract, the cell extract has got a lot of debris. It will have a cell membrane, that is the fragments of the cell membrane. it will have organelles, it will have a cytoplasm, it will have the DNA, it will have the RNA, and it will also have the lot of protein molecule. It means that if you take the cell extract has got many more different types of the biomolecules and the debris apart from the DNA molecule. That's why what we have to do in this particular step, we are supposed to purify the DNA from the rest of the materials. That is the very important point. And next important point is the concentration of the DNA sample. Sometimes once you extract the DNA molecule, the extracted DNA molecule may be pure, may be concentrated, and sometimes you will find that the extracted DNA may not be so concentrated, it is very dilute. Then what you have to do, you have to go for a concentration step, and once the concentration is done, then you will find that you will get a desired type of the DNA sample will be available to you for the next set of experiment. And that's why these are the 5 different types of the steps which are there. Now after that, what is the important? Now we have extracted DNA molecule, we have concentrated the DNA molecule. Now we have to study the quality of the DNA molecule. What is the quality, whether it is suitable for the further studies or it is not suitable, that we have to decide. And that is done by means of a qualitative analysis of the DNA. Many procedures are there. We use agarose gel electrophoresis, we use a spectrophotometer. By using both the techniques, we can actually go for the qualitative analysis of the DNA molecule. The second important thing is the quantity of the DNA molecule. What is the quantity of DNA molecule? You should have at least 50 micrograms of DNA per ml, then only it will be used for the other experiments. And if the DNA is very less, then that particular sample may not be of any use. That's why we have to decide what is the quantity of the DNA molecule, what is the total amount of DNA present in a per ml of that particular sample. That is also very important, and that is also done by means of a technique of the that is the spectrophotometer, as well as the agarose gel electrophoresis. Both of them can be used for the analysis of the quality as well as quantity of the DNA molecule. Okay, the next is now we are going to discuss all these things in detail. The first step is called the tissue preparation or growing and harvesting of the bacterial culture. Now, the first one is if you want to extract the genomic DNA, then what you have to do, you have to select the a particular type of the cell or the tissue from which you want to extract the DNA molecule. The first type of the most of the time what we do, if we are working on human beings, we generally prefer the WBCs, because WBCs are the nucleated cells, and we can easily culture the WBCs in the laboratory, so that we can get a good cell count. All those things can be done. That's why in fact what we do, we generally take the nucleated blood cell, that is the WBCs, because they have a nucleus, and since they have nucleus, within the nucleus the DNA will be present. That's why we prefer the, otherwise if you take the RBC, we can't use RBC, because you might be knowing that in majority of the that is the mammals, including the human beings, you will find that the RBCs do not have any nucleus. It means that they don't have any DNA molecule. And that's why out of different types of the blood cell, we prefer the WBC, because it has got a nucleus, it has got a DNA. That's why this is the one very favorite type of the cell which is used in case of the human beings for the extraction of the DNA molecule. The procedure is also very simple, and without any problem, without any too much difficulty, you can extract the DNA from the WBCs of the human being. That's why it is one of the most preferred type of the cell which is used for the extraction of the DNA molecule. And sometimes what we do, we have to take the DNA from the some solid tissue. Take for example, if you are working on some invertebrates, and you may have to take the DNA from the some solid tissue, maybe from the epithelial tissue, maybe from some other that is maybe from some very muscle tissue, or maybe from the foot or some other tissue. That's why maybe from the gills, or all those things will be there. That's why this is called the solid tissue. You can use any solid tissue. It can be a muscle mass, it can be a foot, it can be a mantle tissue, it can be anything actually. You can even extract DNA from the gills also. It means that any solid tissue can also be used. The only thing is that when you compare the extraction of DNA from the WBC and extraction of DNA from other solid tissue, the extraction procedure is slightly difficult in case of the solid tissues, whereas in case of the WBCs, the extraction of the DNA molecule is very easy. And when we use the solid tissue, we use the solid tissue only when we are not available with the nucleated blood cells actually. Then we have to go for the solid tissues only. Okay. And third type of the material which is used for the extraction of DNA is called as the hair follicles. You might be knowing that even if you have got a single hair, Even from the single layer also you can extract the DNA molecule. It is generally done in case of the forensic investigations. In case of criminal cases related with some crimes, you might have heard about the collection of the samples like hair, or they take the that is the blood samples, or they take the epithelial cells actually. And whatever these particular cells are there, like hair follicles or the epithelial cells, all these cells can also be used for the extraction of the DNA molecule. That's why this is also another very important type of the source which can be used for the extraction of the DNA molecule. And next one is that sometimes what we do, we have to extract the genomic DNA from the bacteria. In case of the genetic engineering, it is a very common practice that we should use so many different types of the bacteria, and in many of the times we have to extract the DNA from the bacteria. That's why we can find that the extraction of the DNA from the bacteria is also very important. And in case of this particular bacteria, what we do, we take this particular bacteria and we grow them in the media. And there are two different types of the media are there. One is called the defined media, and second is called the undefined media. One of the most common type of the defined media is called the M9 media. Defined means everything is known. You know the each and every composition of that particular media. It is called the defined media. That's why we call it as a defined media. Undefined media means we don't know anything about that. What is the composition? We don't know. And such type of the media is called the undefined media. That is the, you take the LB broth. LB broth is nothing but is an example for the undefined media. And we don't know exactly how much quantity of each and every molecule is present in that particular DNA. That's what it's called. And what is the procedure? We have to grow it in a culture media, maybe for about 24 to 48 hours. And once the culture is grown to a sufficient quantity, and then how to analyze? We have to analyze by using the spectrophotometer, absorbance taken at 600 nanometers. And at this particular nanometer, you can find at 0.8. 1 OD is equal to 0.8 into 10 raised to 9 cells per ml. If you get 0.8, that is the 1 OD, it means that you have got a bacterial culture of about 0.8 into 10 raised to 9 cells per ml. This is the one very important thing. And how much bacteria are needed for the extraction of the DNA molecule? We need somewhere about 2 to 3 into 10 raised to 9 cells per ml. It means that if you get 1 OD is equal to, that is now I will just write over here. If you take 1 OD, okay, is equal to 0.8 cells per ml. It means that if you want to have 2 to 3, it means that if you have got somewhere about 2 or 3 OD, it's more than enough for you to extract the DNA from that particular bacteria. That's why you can find that whenever you use the bacteria, you have to take the spectrophotometric reading at 600 nanometer, and 1 OD corresponds to 0.8 into 10 raised to 9 cells per ml. And how much bacteria are needed? You need somewhere about 2 to 3 into 10 raised to 9 cells per ml. This is the number of bacteria which are required for the good extraction of the DNA molecule. Then we also use the plant tissues. Sometimes if you are working on the plant recombinant DNA technology, then we have to extract the DNA from the plant tissue. And I think you might be knowing that plant tissues are much more, that is the harder to break open. That's why we have to use some other technique. That's why we can find that this we are for this particular plant tissue, generally we treat the plant material with the liquid nitrogen. That will help in the breaking up of the cells actually. Then next one is the preparation of the cell extract. How we prepare the cell extract? And in order to prepare I already have said that what is preparation of the cell extract? The preparation of the cell extract is nothing but it is we have to lyse the cells so that the content should get released into the media. And whatever that particular process is there, the lysis of the cell to release the content is called as then we will get the cell extract actually. And whatever this particular lysis of the cell is there, generally it is done by means of two methods. One is called as the physical method, and second one is called as the chemical method. In case of the physical method, what we do, we use the liquid nitrogen. Already I have said that if you are working with the plant material, then it may not be possible for it to extract DNA by means of the soft techniques. You have to go for a techniques which are very harsher. You have to use the grinder and motor And in that, what you are going to do, you are going to use the liquid nitrogen. And once you treat this particular liquid nitrogen, then it becomes very easy for you to break the cells open. That's why I can find that this is one of the very important type of the techniques which is used in case of the plants. But this technique is not at all used in case of the bacteria as well as in case of the animals. In case of the bacteria and animals, what we do, we use the chemical agents. So many different types of the chemical agents are there. These chemical agents can be used. One method is the use of the C-tab, and whatever the C-tab is there, the C-tab method can also be used. And by using this particular C-tab, what we are going to do is that we are going to open the cell and we can release the content of that particular cell. This is also possible. And what is C-tab? The C-tab is nothing but the cityl trimethyl ammonium bromide. The long form of the C-tab is the cityl trimethyl ammonium bromide. And whatever this particular chemical is there, most of the time you can find that this particular technique is generally not preferred in case of the extraction of the DNA from the animals or from the bacteria. It is also used for the extraction of DNA from the plant cell. Then what are the techniques which are used for the bacteria? We use the enzyme called the lysozyme. And I think you might be knowing that the lysosomes are very common in case of the tears. Whenever you take your tears, the tears are very rich with the lysosome. You take the saliva is also very rich with the lysosome. And you take the egg, the egg is also very rich with the lysosome actually. And what is the action of the lysozymes? The lysozymes generally work as the they have exhibit the muramidase activity actually. And what they do, they actually break open the polypeptides which are present in case of the cell wall of the bacteria. Because you might be knowing that in case of the bacteria, the cell membrane is covered by means of another very tough envelope, and that particular tough envelope is called the cell wall. And the cell wall is made up of a very structural, very rigid type of the structural polysaccharide. And in order to break that particular structure polysaccharide, we use the enzyme called the lysozyme. And the lysozyme has got a muramidase type of the activity. That's why these particular enzymes are very useful for the to break open the cell wall. This is the function which is done by means of the lysozyme. Then what we do, we use another type of the that is the mixture, and that is called the lysis buffer. Generally, the lysis buffer contains SDS and the EDTA, that is the sodium disulfide and ethylene diamine tetraacetate. These are the two important chemicals mixed together. And whatever the lysis buffer is there, the lysis buffer is the one of the most common type of the method which is used for the extraction of the DNA from the animal cells. If you are working on the animal cell, maybe from the WBCs or from the some other cells, maybe from the epithelial cells, maybe from the hair follicles, you prefer this particular technique, what we call it as a lysis buffer. The lysis buffer only has got two chemical. One is the SDS, second one is EDTA. Both of them are very important type of the chemicals which are mainly responsible for the disruption of the cell membrane. What the SDS do? The SDS will actually it will remove the lipids from the cell membrane. I think you might be knowing that if you take the plasma membrane has got a important component of the plasma membrane is the lipid bilayer. And once that particular lipids are destroyed or removed, then automatically the integrity of that particular membrane is lost. And once the integrity of that particular membrane is lost, automatically you can find that the content get released. And another type of the chemical we use, that is called the EDTA. What is the function of the EDTA? The EDTA generally removes the magnesium from the cell membrane. What it does, it is not going to affect anything else. It is only going to remove the magnesium. And we know that the magnesium is also very important for the integrity of the cell membrane. And if the magnesium is removed from the cell membrane, automatically you can find that it will completely destroy the integrity of the plasma membrane or the cell membrane. That's why these two chemicals are very important type of chemicals which are generally used in case of this thing. And if you take EDTA, one more important function of the EDTA is that EDTA also suppresses the activity of the DNases. Different types of the enzymes are there. These enzymes can actually go and damage the DNA actually. And that's why in order to prevent the damage to DNA, you can find that this particular enzyme can also be used to suppress the activity of the DNase, like that is the DNA degrading enzymes. That's why this is also very important. And sometimes what we do, we take the combination of the lysozymes and we take with the SDS, because whenever you use the SDS and the SDS along with the lysozyme, the effect of that particular lysis buffer will be very good. And sometimes what we should not use, and one more important thing is that we should not use the lysozymes with the EDTA, because EDTA is a inhibitor of the lysozyme actually. That's why, since the EDTA inhibits the functioning of this particular enzyme, that's why we should not use the combination of the lysozyme with the EDTA, but we can use the lysozymes with the SDS. That's why I can find that Generally, the lysis buffer for the animal will have a SDS plus CDTA, and for the lysis of the bacterial cell, what we do, we use the combination of the lysozyme along with the SDS. These are the two different types of the combinations which are generally used for the lysis of the DNA molecule. Next is the purification of the DNA. Now, whatever the cell extract is there, it has got a lot of material. It has got cell debris, because what we have done in the cell extraction, we have only lysed the cell. We have got fragments of the cell membrane. We have got a protein. We have got a DNA, and we have got a RNA. And what we have to do is that we have to remove all these materials, and we have to only retain the DNA. Then only we are going to get the pure DNA molecule. Otherwise, we will have a lot of impurities. And one very important thing for the removal of the that is the debris is nothing but just, since the debris will have a very larger fragments, and that's why what you have to do is that you have to centrifuge. And once you go for a centrifugation of about 30 minutes, what you are going to do is that you are going to get rid of all the debris. That once the debris are removed, the cell extract will only have three materials. One is the protein. You will get a clear supernatant, and once if you take that particular supernatant, you will have a proteins, and you will have a DNA in that, and you will have a RNA. These are the three important type of the biomolecules which are present in case of the cell extract. Now, what we have to do, the first step is to remove the proteins. We have to remove the protein, and in order to remove this particular protein, what you have to do, we have to go for a particular step, and that particular step is called the deproteination. Deproteination is nothing but it is the removal of the proteins is called as the deproteination. Deproteination is nothing but it is the process of removal of the protein is called as the deproteination. The name itself gives a meaning. There is no problem in that. And then what we have to do, we have to go for, there are two important methods. One is called the phenol method. You can take maybe somewhere about 1 ml or 2 ml of the phenol and treat your cell extract with the phenol. Then what will happen, the phenol will degrade the protein, and it will remove it by means of a that is as a coagulated material. That is the one method. And if you want more effective technique, then what you can do, you can go for a phenol chloroform method. When you take the phenol chloroform method, the phenol chloroform method is much more efficient than the phenol method. And what we have to do is that you have to take 1:1 quantity of the phenol and chloroform, and whatever this particular phenol and chloroform is there, this will actually give the that is the give the better results than the phenol method. Actually, this is the first step which is generally used for the removal of the, in order to go for the removal of the protein from the cell extract. The second important step is we have to use the enzymes. And what type of enzymes we use? I think you might be knowing that there are enzymes which can selectively degrade the protein, and such type of enzymes are called as the proteolytic enzyme. And one very common example is called as the proteinase K. If you take the proteinase K, the proteinase K is one of the very powerful enzyme, and what it does, it actually selectively degrades the protein molecule, and it will remove the protein molecule. That's why we can use the two steps. Now, which one is to be used first and which one is to be used later, that is also very important. Sometimes if you go for phenol chloroform method, a single step of phenol chloroform method may not be enough for you to remove the protein, because protein is present in large quantity. And since the protein is present in large quantity, in very massive proteins are present, that's why a single treatment of chloroform, of phenol chloroform of the phenol method may not be suitable. That's why you have to go for the repeated two to three times of repetition of this particular procedure. And then what will happen, as a consequence of this particular thing, every method of every treatment of the phenol chloroform is followed by means of a centrifugation. And what will happen, if you centrifuge the DNA repeatedly, the DNA molecule is going to be broken into this pieces actually. That's why what we have to do, we have to go for the enzymes, treatment of the enzymes first. And what it will do, it will convert the larger proteins into small peptides. And once that is done, then what you have to do, you have to use this particular phenol chloroform method after the enzyme treatment. Then what will it will do, it will efficiently remove the proteins from that particular cell extract without damaging the DNA molecule. You need not have to repeat the centrifugation. In a single centrifugation, you can complete the removal of the protein. That is, the deproteinization can be done by means of a single step. That is a very important. And then this is the diagram of this particular thing. Here you can see that this is the cell extract we have, and then we mix this particular cell extract either with the phenol or with the phenol chloroform. Then we go for a centrifugation, and once the centrifugation is done, then we get two clear zones. Here you can see that this is the organic zone. Here you can find that this is the organic zone will be there. Here you will get the phenol and chloroform will be there. And here you can see that there is an interface. Here the interface between these two liquids. This is the another liquid. And what is this liquid? This is the aqueous layer, and in the aqueous layer you will have both DNA as well as the RNA, and whatever this particular interface, at the interface of the aqueous layer and the organic layer, you will get one particular white precipitate. And what is this particular white precipitate? This white precipitate is nothing but it is the coagulated proteins. Once the proteins are degraded, that is the degraded, then you can find the degraded by means of the phenol, then all the proteins will get and settle at this particular region, and that's why you can find that once this centrifugation is done, then you can find that you get aqueous solution. This aqueous solution will have a DNA and RNA, and you will have an organic solution, and at this particular center you will get the protein. Now what you have to do, you have to take this particular aqueous layer from this particular tube, and you have to separate it. This is very, now once you have done that, what you are going to get, you will get a partially purified cell extract. What is, why we call it is a partially purified cell extract? Because out of three different types of the biomolecule, that is the proteins, DNA and RNA, we have successfully removed the protein molecules, and still that particular cell extract has got a DNA as well as the RNA. It has got DNA as well as the RNA. That's why we call it as a partially purified cell extract. Now what we have to do is that the next step is to separate the DNA from the RNA molecule. That is also very, very important. And in this particular purification of the DNA from the RNA is generally done by means of one enzyme, and that particular enzyme is called as the RNases. You take there are specific enzymes called as the RNases. What they do, they actually convert the RNAs into small oligopeptides, and that's why once the small oligopeptides are there, the DNA will be very, very large. It gets settled when you go for a centrifugation after treatment with the RNAs. What you have to do, you have to go for the centrifugation, and once you go for a centrifugation, then what will happen, the DNA gets settled as a pellet at the bottom of the tube, and whatever the RNA is there, the RNA will be present in case of the supernatant actually. That's why, since it has been converted into the oligo small peptides or the oligonucleotides, that's why the removal of that particular RNA from that particular cell extract becomes easy, just by means of treating that particular cell extract with the RNAs at 37 degrees. This is very, very important, because the RNAs is very, very sensitive to the temperature, and if the temperature is less or more than 37 degrees centigrade, that will actually interfere with the functioning of this particular enzyme. That's why precisely you have to use the 37 degrees centigrade, then the efficiency of this particular enzyme will be very, very good. That's why just treat that particular partially purified the cell extract with the RNAs for about 5 to 6 hours. Then what will happen, centrifuge it, and once you centrifuge it, you will get this is the small fragment oligonucleotides of the RNA, and at the bottom, because of this particular image, you are not able to see the pellet. At the bottom you can actually see the pellet. In this way you can find that. Now what we have done, we have removed the proteins and we have removed the RNA molecules. With this we can find that whatever that particular solution is there, that particular pellet will have that is the pure DNA molecule. And if once the DNA is completely purified, then what we have to do, we have to see what is the concentration of the DNA molecule. I already said that for any recombinant DNA technology experiment, if you want to go for any recombinant DNA technology experimentation, maybe any experiment, it may be PCR, it may be something else, cloning or something else, you have to have a minimum quantity of the DNA molecule. And if the DNA is already concentrated, you need not have to go for any concentration step. If the DNA itself, you just by means of the viscosity, you can judge whether the sample is a concentrated DNA or a dilute DNA just by means of the observation. No need of any machine, nothing is required. Just by means of the observing the viscosity in the tube itself, you can judge whether the given that is the DNA is concentrated or it's a diluted. If the DNA is concentrated, then there is no need for any concentration step, and if the DNA is dilute, then you have to go for a concentration step. And how we do the concentration, this is called the ethanol precipitation. This particular concentration is called as the ethanol precipitation. Most of the time we use the alcohol called the ethanol. Sometimes you can also use the isopropanol, can also be used for the concentration of the DNA molecule. And you have to carry out this particular ethanol precipitation at minus 20 degrees centigrade, maybe for about 6 to 8 hours. This is the minimum duration of the treatment. And if you prolong it, then it is good actually. You can even keep for a day or two also. If you are in urgency of conducting the rest of the experiment, then you can use it in 6 to 8 hours. But if you don't have any urgency, You can, if you want to use it later, then what you have to prefer is that you have to concentrate the DNA maybe for about 1 or 2 days at minus 20 degrees centigrade. And please remember that along with the ethanol, you must also use a very small quantity of the NaCl. This is also very, very important. And then you will get the, once this particular procedure is completed, then you will get a very, very good concentrated DNA will be there. And that particular concentrated DNA can be used for the other experimentation in future. You can preserve it. If you want to use it immediately, you can use it immediately. Or if you want to preserve it for a future use, you can keep it in a minus 20 degrees centigrade for a long time. Nothing will happen to that particular DNA. It can be kept for hundreds of years. Nothing will happen to the DNA, because we already discussed in nucleic acid that DNA is a very, very stable molecule, and even at room temperature you can keep it for a very, very long time. That's why there is no problem with the DNA molecule. And this is just a flow chart of the extraction of the DNA molecule. Here, same thing, whatever you have discussed, all those points we have representing it in a flow chart. And this is the diagrammatic representation of the extraction of the DNA from the genomic DNA. And now we are going to discuss about the qualitative and quantitative analysis of the DNA molecule. I already said that once the DNA is extracted, we have taken maximum precaution to remove the protein. You have taken the maximum precaution to remove the RNAs. But what is the guarantee of that we have removed everything? That's why we have to go for some scientific methodologies, and by using that particular techniques, we can analyze whether the extracted DNA molecule is of desired quality or it is not of desired quality. What we have to do, there are two important techniques are there. One is called the agarose gel electrophoresis, and second is called the spectrophotometry. You can use both for the qualitative, that is in order to judge the quality of the DNA molecule as well as the quantity of the DNA molecule. You can use the agarose gel electrophoresis, and even for the quality, even by using the spectrophotometer, you can actually judge the quality and quantity of the DNA molecule. That's why both these techniques can be used for the to judge the quality and quantity of the DNA molecule. Now we will discuss one by one. Generally, for the genomic DNA molecule, we use the 1% agarose, because that particular pore size should be larger enough to allow the DNA to go, DNA to pass. That's why 1% agarose is generally used. Here we can see that if you take this particular diagram, we can see that we are going to discuss about the that is the quality of the DNA molecule. Now you can see that if you take this particular figure, here you can see that this is one genomic DNA molecule. This is another genomic DNA molecule. This is the third DNA molecule. If you see this particular here you can see that there is a discrete band is present, and whatever this particular discrete band is there, it is present very, very near to the loading well. This is the loading well. These are the, you can see the that is the dark lines here, and these dark lines are nothing but they are the loading wells actually. And here, once you have loaded that particular DNA molecule and you have run the gel electrophoresis and for a considerable period of time, and then you can see that there is a discrete band which is present very, very near to this particular. It means that this particular DNA is very, very large, and that is the quantity of DNA. If you take the size of this particular band, you can, if the band is very thick, it means that it is a good quantity of DNA is present. And if this particular DNA is present very, very near to this particular well, it means that it is a of very, very good quality DNA molecule. Now we will see the second picture. Now here you can see that there are two, this you can see that now you can't see discrete band actually. Here if you take this particular thing, this is also genomic DNA. The only thing is that here you don't see any discrete band. It means that whatever that particular DNA is there, it is broken into small pieces, and that's why we can actually see the sharing of the DNA molecule. This is the shear of the DNA molecule, small fragments, and they run at different speeds, and that's why we get this particular that is the smear like pattern will be there, and this is the sheared DNA. It means that the DNA is completely broken into pieces, and whatever this particular DNA is there, it is of no use in recombinant DNA technology. We can't use this type of the DNA. It is very, very poor quality, and it is not at all suitable for the experimentation in the recombinant DNA technology. And if you take this particular step, here you can see that the DNA is not very, very, if you take the size of the band, band is slightly discrete, but only thing is that band size is very small. And second thing is that if you see just above this particular, you can see some white smear, and this particular white smear is nothing but is the presence of the RNA molecule. It means that here there is a RNA molecule is present. Here you can find that the DNA is broken into small pieces. That's why these both the DNAs are not at all suitable. Whereas if you take this particular DNA, here you can see that there is a discrete band is present, and because of the discrete band, you can actually, this is the good quality of DNA. The second method is called as the spectrophotometric technique. And what we do, we take readings at 260, absorbance at 260, And what is the inference? If you get one OD, it means that the DNA sample has got a 50 micrograms of DNA per ml. This is the one important parameter. Here what we are going to judge? We are going to judge the quantity of the DNA molecule. One OD corresponds to 50 micrograms of DNA per ml. Then how to judge the quality of the DNA molecule? You have to take absorbance at 260 and absorbance at 280. And if you take the ratio should be 1.8. If you get a value of 1.8, it means that it is a very, very good quality of the DNA. That's why by using both hydrogel electrophoresis as well as the spectrophotometer, we can actually analyze the quality as well as quantity of the DNA molecule. And with this, we are going to stop this video. And in the next video, we'll discuss something, some other aspect related to the recombinant DNA. Thank you very much for your patience.